So far in our discussion of metabolism, we've been focusing primarily on the molecule glucose or other monosaccharides that can be very easily shifted into or converted to glucose. Now, during times when we are well fed, there is more glucose in our blood than what our body actually needs at that particular time. So in order to manage the amount of glucose in our blood, our liver and skeletal muscles will take extra glucose and generate a molecule called glycogen. Now, we've talked a little bit about glycogen. I showed you guys the structure of glycogen in chapter 15 to help you guys become aware of that. It is a chain or a network of glucose molecules that are linked together by an alpha 1 to 4 glycosidic bond. Now, the importance of glycogen comes into play because glycogen is a very important storage place for glucose. Now, glucose is the sole source of energy for several very important cells within the mammalian body. So red blood cells and the central nervous system, particularly the brain, utilize glucose as almost exclusively as their source of energy. Now, generally, when we're in a well-fed, continually well-fed place, then the diet is obviously going to be a suitable place for the red blood cells in our brain to acquire glucose. However, during bouts of food deprivation, whether that be a relatively short period of time, like 5 or 6 hours, or whether it's for an extended period of time, our body has to replenish glucose back into the blood in order for the red blood cells and our brain to utilize for energy. Now, one major place where the body is going to look to accomplish that is going to be in the breakdown of this molecule glycogen. So we're going to be thinking about glycogen in this mini lecture and how it's synthesized during times of plenty and how it's degraded during times when your body needs to generate more glucose for the blood. We'll then a little bit later talk about this process called gluconeogenesis, where your body uses non-carbohydrate molecules to create glucose for the purpose of getting glucose levels in the blood up to the point where red blood cells and the brain can actually benefit from those. So your book utilizes the term glycogenesis. I tend to just refer to the metabolism of glycogen either by glycogen synthesis or glycogen degradation. So those are the two ways that you want to be familiar with the processes that we're going to talk about. So glycogen synthesis is going to occur when we have plenty of glucose available. So it's going to store glucose in a polysaccharide form so that it can be readily available at later times. So what happens is when there is a large amount of glucose available and the glucose has been brought into cells, the glucose is going to be converted into glucose 6-phosphate, which we talked about as being the first step in glycolysis. Well, when glucose 6-phosphate levels are particularly high, then the cells of our liver and the cells of our skeletal muscle will interpret that as being in a very well-fed condition. And so those organs will begin to shift into storing that glucose for later. Now, the key here is that there's only going to be a limited amount of glycogen that the body is going to store. The body isn't going to store excessive amounts of glycogen because it's actually a kind of a large molecule and really a little bit tricky to store in the long term. So up to a certain level, the body will store energy as glycogen, but then it'll shift into storing energy in the form of lipids, which we'll talk about in chapter 17. So the key in managing body weight is going to be the balance of caloric intake to caloric output. And what that means is that you should only take in the amount of calories in food that you are able to expend during a particular day. Now, that's because regardless of what form your body gets energy, whether it's all carbohydrates or all fats, it doesn't matter what form your body receives that energy. Your body will only store a certain amount of that energy as glycogen or carbohydrate stores, and the rest of it will convert to fat. Again, our bodies are made to survive, and storing energy as fat is a more efficient way to store energy. And again, we'll talk about that in chapter 17. So there's going to be, whenever we are in a well-fed state, our body's glands will secrete hormones that will indicate that there is ample glucose and that extra glucose can be stored as glycogen. Now that hormone is insulin. So insulin stimulates the synthesis of glycogen. So I don't want you to get too alarmed by the diagram that you see here on this slide. I'm not going to have you guys worry so much about the nitty-gritty details of glycogen synthesis and breakdown as much as understanding conceptually when glycogen is synthesized and why, and when glycogen is broken down and why. So understanding those concepts and what plays in to that, to each of those events, is really what I want you guys to focus on. Now I will talk a little bit about the steps that lead to glycogen formation, but again, don't get bogged down in the details as much as the bigger picture of what's happening. So glucose, when it's brought into the cells, it's brought in as pure glucose. Now once inside the cell, hexokinase, the enzyme that you learned about in the previous lecture, will convert glucose to glucose 6-phosphate. Now once glucose 6-phosphate is present, or that once glucose is converted to glucose 6-phosphate, it locks that glucose molecule into the cells of our body, and most of the cells will then keep that glucose molecule. Now when there's extra glucose around, the cells of our liver and skeletal muscle can take extra glucose and store it as glycogen. Now in order for that to happen, there's going to be an isomerization that occurs where the phosphate is going to be relocated from carbon 6 to carbon 1. So again, that's going to be converted by an enzyme called a mutase. So the glucose molecule goes to glucose 1-phosphate. The glucose 1-phosphate is then going to utilize one energy equivalent. So the UTP is going to be energy equivalent to ATP. So this is an energy requiring process. So a UTP molecule is going to be used and added to the glucose molecule. And then this glucose UDP molecule is going to be what is used to add one glucose molecule at a time onto the growing end of a glycogen chain. So glucose UDP plus the glycogen molecule that's already present is going to result in the glycogen molecule being one glucose molecule longer than it was previously, and then the UDP is going to be released. Now that UDP can be converted back to UTP to prepare another glucose molecule to be added on to the glycogen chain. So this is glycogen synthesis. So glycogen degradation is going to be the opposite. It's going to be the process by which glycogen is, whereby glucose molecules are released one at a time from the larger glycogen molecule. So by breaking it down, the glucose, the body is, or the cells of either the liver or the skeletal muscle are providing glucose for use in energy, or for energy production. Now skeletal muscles are a bit on the selfish side, so any glycogen that's stored in the muscles will provide glucose molecules only for muscle, whereas the liver will provide glucose molecules for all of the tissues of the body. And we'll talk more about that in a little bit. Now, when would this be necessary? Well, it's going to be necessary during times of food deprivation. So when glucose levels begin to decrease in our blood, the body will provide a way to get those glucose levels back up. Because remember, red blood cells and the cells of our brain absolutely require glucose for their normal function. So during food deprivation is going to be a very important time for your liver to provide glucose back into your blood. Now, the other possibility is going to be under conditions of stress or threat. And we'll talk about how each of these works in the next couple of slides. So glycogen degradation, or the breakdown of glycogen into individual glucose molecules, is going to be controlled by two hormones. The first is the hormone glucagon. So glucagon is going to be secreted in an opposite manner, for the most part, in an opposite manner to insulin. So whereas insulin is secreted when the blood glucose levels are high, glucagon is secreted when blood glucose levels get low. So when the blood glucose levels drop, the pancreas will respond by secreting glucagon. Now, glucagon will be a signal to your liver that there's a need for glucose to be generated and to be added back into the blood for the use for the red blood cells and for the brain. Now, the other possibility is going to be the molecule epinephrine or adrenaline. You guys probably know that term or know that hormone as adrenaline. So adrenaline is going to be, or epinephrine is the hormone that is related to the fight or flight response. So times of stress or threat are also going to be times when we want ample glucose available in the blood, because we want to be able to respond to whatever that threat or stress is. Now, granted, when I think about the fight or flight mechanism, I think about skeletal muscle and the importance of movement to respond to that stress. So our skeletal muscle will need glucose. And so epinephrine is a way to make sure that glucose is put back into the blood and made available for the skeletal muscles in a way to respond to that stress. So, glycogen is going to be synthesized in response to insulin during times of high glucose availability. Again, it's going to require ATP and UTP for that to happen. And again, all of this is going to be activated by the hormone insulin, which results in glycogen synthesis. Now, this process of taking glycogen and taking glucose and converting it into glycogen is going to be inhibited by both glycogen and epinephrine. So these hormones are basically regulating the process in opposite ways. Now, during times of low blood glucose, our glucagon levels will increase, or during times of threat, our epinephrine levels will increase, and these will signal to the liver or to the muscle that glycogen needs to be utilized to generate glucose. So the degradation of glycogen is going to be activated by both glucagon and epinephrine, but inhibited by insulin, so that these two processes aren't working against each other simultaneously. Now, when glycogen is broken down, it's broken down into the form of glucose 1-phosphate. So that this glucose 1-phosphate can then be converted into glucose 6-phosphate. Now, glucose 6-phosphate can then enter the glycolytic pathway. So glucose 1-phosphate being converted into glucose 6-phosphate is going to be activated by insulin. It's going to encourage our bodies to take this glucose 6-phosphate and utilize that in the process of glycolysis to generate high energy molecules. So glucose 6-phosphate, as I mentioned earlier, is a form of glucose that is trapped inside of cells. So when a glucose molecule is in your brain or your skeletal muscle, the cells of your brain or your skeletal muscle, then the glucose 6-phosphate molecule isn't going to go all the way back to glucose, because the brain and the skeletal muscle cells will actually utilize this glucose 6-phosphate in the process of glycolysis. However, your liver contains an enzyme that will allow the conversion of your glucose 6-phosphate molecules all the way back to glucose. So it removes a phosphate. Now the enzyme that does that is called glucose 6-phosphatase. So phosphatases remove phosphate groups, and in this particular case, the cells of the liver will remove a phosphate from glucose 6-phosphate to get us back to glucose. Now this glucose molecule can then leave the liver, and it can also happen in the kidneys, but it can leave primarily the liver and enter back into the blood, and once it's in the blood, it can circulate through the body, and any tissue of your body, especially your brain or red blood cells, can take that glucose and utilize it for its own energy needs. So there's always going to be a balance between glycogen synthesis and glycogen degradation. Now, during times of high blood sugar, we also refer to that as hyperglycemia, insulin will stimulate cells to take in glucose and either utilize that glucose in glycolysis or store that glucose in the form of glycogen. So insulin will activate enzymes that will use glucose for energy, so the first step of glycolysis, or it'll activate certain cells to store that glucose as glycogen. At the same time, insulin is going to inhibit the degradation of glycogen. We don't want to be trying to build something at the same time we're trying to break something down. Now the opposite is going to happen during times of low blood sugar or hypoglycemia. During times of low blood sugar, glycogen is going to stimulate the breakdown of, glucagon is going to stimulate the breakdown of glycogen to provide glucose for energy within the body. So at the same time that it's stimulating the degradation of glycogen, it's inhibiting the synthesis of glycogen. So again, these hormones are working in opposite manners to regulate whether glycogen is being made or whether glycogen is being broken down. So this is a really wonderful example of how our body is being a good steward of the energy molecules that are available. When there's a lot of energy molecules available, then we'll utilize the energy in glycolysis or we store it for later. However, whenever we get to the point where there's a low availability of that glucose molecule, then our body looks to those stores that were previously made for energy. And again, the liver is going to be instrumental in keeping our blood glucose levels at a normal, stable level so that our brain and our red blood cells can function normally as they should. Now we'll talk a little bit more about this process of balance when we think about how all of the metabolic pathways are integrated and how our bodies kind of work in a collective way to make sure that the energy needs of all tissues of the body are met. But for now, we'll stop and you guys can try to make some sense of all of this and try to capture the big picture of what's happening with glycogen during times of plenty and during times of need.